as we saw here that 17 kDa fluorescent labeled dextrin shows strong binding to the cell surface. So this gives us the hint whether we can use this phenomenon to gate certain smaller small molecules. And if you do the confocal scanning, this is just one individual beads. If we scan this from the different optical sections along z-axis, we saw every layer you saw very strong binding. So we tested using 55 more DNA, also siRNA. Both of them can internalize, but after watching, they are still retaining certain with cell within the wall. And we are sequentially incubate with siRNA and 17 kDa dextrin. We saw that this can make the siRNA retain in the interior cavity, suggesting that this has potential as carrier for siRNA. siRNA is everyone knows that is very widely used reagent nowadays, but there is a challenge how to deliver this successfully. It's a challenge. We also found that the internalized siRNA by using this strategy can release, the conserved release. So next question we ask, can PWFGMs be a carrier for protein? We have tested different size of proteins from 13.7, 29, 43, and 75 to 150. 150 is a regular IgG antibodies. And we found that 75 kilodalton protein and antibody can internalize. And they are, after rotting, they are still within the interior cavity. And interestingly, this internalized antibody can release gradually. We have using the data vision microscope to observe fluorescence density in every 2 hours. We found that the release speed followed first order kinetics with half-life about 6 hours. So next, we want to test whether we may really use this to deliver the real therapeutic protein, whether we test in the real situation whether it's working or not. So in our laboratory, we are in the process of developing a therapeutic antibody. It's called single antibody. We conjugate this antibody to the folate for the purpose of the receptor-mediated delivery. We are conjugate this protein to the FITs for the purpose of visualization. As you can see here that this glass beads, this single antibody can internalize to the glass beads. They have very strong fluorescence. This is bright field. This emerge. And the next question is that the released protein, whether they are functional or not. So we incubate the loaded beads to the folate receptor cancer cells. We found that the single antibody can bind to the cancer cell surface, can be internalized, which is suggesting that the delivered protein is still functional. So the next question is that whether we can use this carrier for the deliver something to the tumors. So we perform another experiment. We first loaded our fluorescence FITG label dextrin glass beads to the aqueous gel, and we loaded different amount. We performed the quantification. We saw that the amount that loaded shows a linear correlation with photon counts. So that means that the system is working. So once we obtain this data, we inject directly to the tumor site, and we saw under the machine, we saw that there is a strong fluorescent signal suggesting that the beads still retain within the tumor. So this suggesting that this porous hologlass microsphere has potential to deliver something to the tumors. So in conclusion, so the PWFGMs are novel form of glass material consisting of the hollow central cavity with 1 micron thick wall, which has a tortuous network of nanoscale channels. We saw that the porous wall of the PWFGMs promote the size-dependent uptake biological molecules in the 3 to 5 to 8 nanometers range. So these include the antibodies and modified our thinking antibody fragment, and the release product are still functional. And we also saw that 17 kDa dextran can be used to gate porous wall, which can facilitate in control release of the internalized siRNA. And also we saw that dextran loaded in these glass beads remain in their place after mouse tumor injection, suggesting potential application for drug delivery. So the data I present suggests that this novel form of the drug carrier has potential for deliver different reagent, either individually or in combination. And also it can also has a potential as a second generation of the radioisotope delivery carrier for the purpose of their cancer treatment. So finally, I would like to thank Dr. Dana. He's here. He guided this project and gave us a lot of help. And we also like to thank our collaborator from the Savannah River National Laboratory. They produced this glass piece for us. And the outside is very just white. I want to give everyone some colorful things. This is where we came from. Well, we came from Augusta, Georgia. This is famous for the Monster Tournament. And this is the building that's where we are working. And thank you for your attention. I'm more than happy to answer any question you have.
Questions, please line up at the microphone and introduce yourself and your affiliation.
Kester, Penn State College of Medicine. Excellent talk. So when you put the siRNA in the glass beads,
right,
do you actually protect those siRNAs from RNases in systemic circulation? So do you truly get protection? Because a lot of different nanotechnologies are being used now for encapsulation of siRNA, but they truly don't protect the siRNA in biological fluids. I mean, have you done that yet?
Not yet. But our protection is that we have the glass shell, also the gated dextran, should be resistant to some harsh physiological conditions like pH or some enzymes. But we haven't tested in the real system, but theoretically, this has that potential. Mm-hmm. Thank you.
Next question. One more question, Sri?
You basically use glass, and the silicon and the silica that we make is basically glass as well. So how do you check the clearance from the body of these big structures?
That's a very good question. I mean, for the glass, it's called bioactive glass. This has been used clinically for many, many years. And the idea is that, for example, this can be used as a scaffold for the bone formation. That's for sure. Clinically has been proven. And also, within the cell, within the body, because the basic component of this material can be part of the bone. And also, if you deliver this to the other part of the body, the macrophage gradually can absorb it. So not like that glass. This can be absorbable. So this, I think, clinically has approved it's okay.